Hi, guys, and welcome to our Diabetes Chat research event. Tonight, we have the Type 1 Grand Challenge as our guest speakers, and we're really looking forward to hearing about the amazing research projects with our speakers tonight. So, should we have a look at what is coming up on our research event this evening? Let's have a look. So welcome along to Diabetes Chat. I am Tom and I'm the founder of Diabetes Chat and host tonight with my fabulous co-host Mary, so she'll be here shortly. We've got our Grand Challenge event tonight. We have our three fantastic speakers coming up soon, our researchers. And we will be hearing from Liam from the Steve Morgan Foundation as well, introducing what is the Steve Morgan Foundation and also talking about the Grand Challenge. So you can go find out more information on the website there to learn more about the Grand Challenge. So go check that out. They're also our researchers, so you can learn more about their research studies, their projects on the website, the Grand Challenge. So go check that out after the event tonight. And again, there are our researchers. We have James, we have Sarah and Victoria, and you can see there the three different research studies projects that we'll be learning and seeing about tonight on our research event. So lots of exciting research coming out tonight on our research event here. So what is coming up on Diabetes Chat tonight? Let's have a look. We have our introduction here now, just telling you guys how to get involved. Then we will welcome Liam from the Steve Morgan Foundation, CEO, and he will talk about the Steve Morgan Foundation and introduce the Grand Challenge to us, which is really exciting. We'll then have a researcher panel chat, a live Q&A, and we've got some questions for our three researchers. But if you guys have any questions, please get them over, and the researchers are here all evening. So please send them over. We can get them live on tonight's event, or they'll stick around in the chat box and they will answer your questions. So please, any questions or comments you have, we'll share that for sure. Then we will have a 20-minute presentation from each researcher. So you can see around 8:30, James will be our first researcher that will come up and he will share his research project. And then there'll be some follow-up questions, hopefully with you guys, the community listening in tonight. And then Sarah will join us. And then to finish, Vicky will also round up. So lots to come over the course of the evening. Like I said, there are our speakers. So we have Liam, James, Sarah, and Vicky. So lots of exciting things to come over the course of the next hour and a half here on Diabetes Chat. This is our fifth research event, and it's an honor that we're here tonight talking about the Type 1 Grand Challenge. So what we're going to do is we're going to learn a little bit more about the Type 1 Grand Challenge, and we're going to learn about the Steve Morgan Foundation. And it is a great honor to welcome Liam onto our panel tonight, who is the CEO of the Steve Morgan Foundation. So we're going to welcome up Liam to the panel. Thank you, Liam Eagleson, for joining us tonight on our research event. Thank you for coming on.
Thanks, Tom. Great to be with you.
No, it's really great, and it's an honor to have you here. And I know tonight you're going to share with everyone a little bit about you, your connection, and obviously go through the Steve Morgan Foundation, what it is, and the Grand Challenge. So I'll hand over to you, and we look forward to learn a little bit about it. And if anyone has any questions, please put it in the chat box, and Liam will be able to get the questions to you, or you can answer it later on. So I'll hand over to you, Liam.
Thank you, Tom. Thank you. Good evening, everyone. It's great to be with you. So just a few words of introduction about myself and the Steve Morgan Foundation, and then the origins of Type 1 Diabetes Grand Challenge. So as Tom said, my name's Liam. I'm somebody who lives with Type 1 diabetes. I've had Type 1 diabetes for just over 9 years. I have a 16-year-old son who was actually diagnosed with Type 1 diabetes almost to the day 12 months before I was. So he was diagnosed when he was 6. He's now 16, in the middle of his GCSE exams. So we're learning what it is to navigate GCSEs with Type 1 diabetes, which is not fun, I have to say. So that's why we have skin in the game, which is not quite the right phrase, is it? I think something more like blood on the table or something. But we have a personal investment and interest in Type 1 diabetes. I'm the CEO of the Steve Morgan Foundation. I've been there for nearly a couple of years. But just before I joined the Steve Morgan Foundation, they made a commitment to invest £50 million into research into looking to find a cure or prevention for type 1 diabetes. So that is a tripartite arrangement between the Steve Morgan Foundation, JDRF UK and Diabetes UK, and the three organisations come together to try and supercharge the desire to make progress in research on type 1 diabetes. So that's the kind of origin of Grand Challenge. Just before I tell you a bit more about the Grand Challenge, just so you understand who the Steve Morgan Foundation is, we're a foundation that's based predominantly in the northwest. You can see that tiny little map on the screen there. It shows you Merseyside, and then a large wave of North Wales, then a little bit of West Cheshire and North Shropshire into the mix as well. That's the kind of geographical focus of the work of the Steve Morgan Foundation, and we support loads of brilliant charities who are just doing fantastic work on the ground, making a difference in people's lives. So our strapline, or our hashtag, if you like, as the foundation, is changing lives for good, and that's both the intention to make a difference in people's lives in the here and now, but also something about that permanent difference that we can make. So as an organisation, we're interested in really trying to get underneath the presenting issues that we see in so many of our communities around poverty and destitution, and deal with some of those systemic issues. So we give away between 10 and 20 million pounds a year to brilliant charities in our region, making a difference to people's lives. So that's much of our bread and butter day-to-day work. But as I said, about 2.5 years ago, the trustees made this mammoth investment of £50 million into type 1 diabetes research. And that's largely because Steve and Sally, who you can see on the screen there, that's Steve Morgan, who has invested a chunk of money into the foundation that enables us to do all of our work. He's married to Sally, and Sally has a son called Hugo, who was diagnosed with type 1 diabetes when he was about 6 as well, a number of years ago. And so they've got that personal experience as well of being people who care for somebody with type 1 diabetes. They understand the frustrations, the challenges, all of the issues of what it means to care for somebody with type 1 diabetes. So a number of years ago, they partnered with JDRF in some smaller grants into research. But as I say, and as it says on the screen there, we made this £50 million investment into research. And that 50 million is split into 3 different pots. First part is looking at replacing beta cells. The second part is looking at the root causes of type 1, and the third part is looking into novel insulins. And so the next few slides just tell you a little bit more about that. But to be honest, I'm not going to dwell on the detail here, because you're going to hear much better from our brilliant researchers who we'll hear from in a moment around these areas. So replacing beta cells is the biggest pot of our £50 million. And that's because we think this is likely to be the place where the opportunity to make a real difference scientifically around the idea of a cure for type 1 will be. But as I said, we're also investing in trying to understand more about the origins of type 1, and particularly around immunotherapies and how they can help slow down the immune attack that creates type 1, or even prevent that in the first place. So another big area of research. We'll hear more about that later. And then novel insulins as well, how we can modify insulin to make it more effective, faster acting, or responsive to glucose, or a type of insulin that can be injected perhaps once a week, and then it does its job over the course of the week for us. So some really exciting areas. We've all heard those kind of promises of a cure in 5 years, a cure in 10 years. So we don't want to hype up the expectation around that, but we do want to say that there is some tremendous work happening in research here in the UK and globally. But our investment into the Type 1 Diabetes Grand Challenge is all about supercharging and trying to move forward the progress that we can make scientifically to move us closer to that day when we get a cure or the prevention of type 1 diabetes. So I'm going to stop talking. I think I'm slightly ahead of time, but that's good news, because it means that Tom can move the thing forward and we can get to hear from the researchers rather than from me. Thanks, Tom.
Thank you so much, Liam. Honestly, thank you for coming on tonight and representing, obviously, the Steve Morgan Foundation, but showing and sharing with the community a little bit about you and your connection to diabetes, and also what is the Steve Morgan Foundation. And it's amazing all the work Steve and Sally and everyone at the team are doing at the Steve Morgan Foundation. And yeah, it's going to be really interesting this evening to hear them three particular research projects with the researchers. So thank you so much for coming on tonight and giving us a quick sort of introduction of the night ahead. So thank you so much, Liam.
Pleasure. Thank you.
Brilliant. Thank you. So guys, if you do have a question for Liam, please put it in the chat box, and you can then hopefully chat with Liam. He'll stick around over the course of the evening. So if you have a question or a comment about the Steve Morgan Foundation or Grand Challenge, head to our website if you are watching on social media right now. So www.diabetieschat.net/Grand Challenge. And in there, you'll be able to connect up with other people in the community, and you can ask questions and chat along. We have over 88 people listening right now across on our platform. So that's really, really great. And it's nice to see that the community are tuned in tonight, and it will be available on demand after tonight. So you can go watch back and share. So if you're on social media, you can watch us on Facebook and Twitter, which is great. And you can share. So please use the hashtag Diabetes Chat, but also use the hashtag Type 1 Grand Challenge, and you can connect up with many people tonight. Let us know what you think of tonight, any questions for Liam or the researchers, and we'll get that to the team. So I'm going to bring up Mary, who is going to host our next section of the evening. Hello to Mary.
Hi, Tom. How are you?
How are you? You all right?
I'm great. Yeah.
Oh, how fantastic to start the night and learning about the Steve Morgan Foundation with Liam. And I think it's just going to be really fascinating chat tonight. And you have the great privilege to host our next section, talking to our fantastic researchers, right?
I do. And you know what? Like I'm trying to dampen down my excitement and not get too giddy, because I recently heard Sally speak, and she used a phrase that is connected to the Grand Challenge, and it just really warmed me. And I'm trying not even to be too excited by saying that, but she said, "Let's make lived experience of type 1 diabetes past experience for all." And I think I actually started to cry in the audience, and I think this is what this is going to do, hopefully, or accelerate for us. So
absolutely.
Yeah.
So let's crack on, and I can't wait. So I'm going to jump off, and I'm going to bring up our three fantastic researchers, and I'm going to join you for the next 15-20 minutes for a little Q&A. And if anyone has any questions, please send them over, and we'll get them over to Mary and the researchers. So over to you, Mary. I'll bring everyone up.
Thanks, Tom. Hi, James. Hi, Vicky. Hi, Sarah. So this is just a high level before we get into the nitty gritty of all your work, and it's to bring a few ideas together to place it all within our minds. So I'm going to ask 3 questions, and I'll ask each question, and then get you guys to answer and come back to the next question then. So the first question is, how do you feel the Grand Challenge will support future care therapies that wouldn't have been without the challenge itself? So I'll start with James, maybe.
I mean, the Type 1 Diabetes Grand Challenge really has been transformative for UK Type 1 research. It's really enabled the type of projects we can set up and run to be much more ambitious. They've been properly resourced, so we can do things with a sort of 5-year timeframe, and take a few risks. Do things that have not been tried before, and take a few risks in trying to move the field forward. And these type of approaches are quite challenging with conventional academic grant funding. So I think the Grand Challenge fills a really important niche in that kind of funding ecosystem in the UK. And of course, the whole scheme and the type of research they're funding is mission driven. There's a clear, we've got to push towards improving the lives of people living with type 1 diabetes. So I think having that very clear mission driven approach is really important as well.
Yeah, thanks. Well, you said they were challenging. Can you say briefly why that traditional research is challenging? Just
what I was meaning is more conventional academic funding mechanisms. So what we would typically do is apply for some money to run a project, and that be reviewed by a panel and what have you. And this still runs in the same way, but the appetite for risk and more ambitious projects is much higher with this funding mechanism. So I think it's probably fair to say that conventional funding mechanisms are quite risk averse. Panels want to see a lot of preliminary data and smaller steps forward. Whereas this is very refreshing because it wants big and bold steps forward. And of course, the Grand Challenge is a large fund and it's funding lots of different projects across lots of different labs and sort of different collaborative networks. And I think the idea is that some of these will hopefully strike home and come through with a really exciting output. But just having that inbuilt ambition in the scheme is really quite refreshing, I think.
Okay, Vicky, what about you then?
Yeah, to echo what James just said, I think what it's done is it's given us the freedom to think disruptively, to think outside the box, which normally we might be more reticent to do. So we're bringing new ideas from other fields. I, for example, am working with engineers who otherwise would never have thought twice about diabetes. We're bringing new technologies, new materials to diabetes, which perhaps we wouldn't have thought of. And I think the other thing that I would add is about the network of researchers who are now focused passionately and wholeheartedly on type 1 diabetes. And that includes the clinicians who are interested in bringing clinical trials to fruition quicker than perhaps would have happened before. So, this is the UK, we've got the NHS. There is no reason why we shouldn't be at the forefront of pulling on that phenomenal infrastructure to power through translation. And I think that this challenge is making us think collaboratively and as a group better than we've ever done before.
Brilliant. Sarah, what do you think?
So I have to say that James and Vicky have said many of the things that I would have said. So I think we're all singing off the same song sheet here. But I think really, as sort of Vicky touched on, it's really bringing researchers together from different disciplines. So the bioengineers that Vicky was talking about, chemists, stem cell biologists, clinicians, transplant experts, immunologists, basic scientists, all of those different people who've got different expertise and value to add to this. And I think when we're working connected together collaboratively, sharing ideas, being bold with our initiatives, I think that's the way that we're going to make those better leaps forward. And we really are starting to access people who wouldn't normally be in the type 1 diabetes fields. And I think that brings a whole fresh set of ideas in. And I'd also sort of reflect a little bit on what James was saying. And I think this kind of funding mechanisms and the teams that we, as the fellows, the Grand Challenge fellows, are able to do, but also now the wider SMF grants that are coming through, is we are getting the opportunity to train and bring on the next generation of scientists who are going to be our future, because we aren't going to be able to solve it overnight. But if we can get those people and the infrastructure in place in the UK in a way that we just haven't been able to do before, I think with all those new fresh brains in the mix as well, we're really looking, the future is bright.
Yeah, thanks. It's very very innovative and it's very powerful, I think. And there's such an energy that you can see that from the collaborations. The second question then is more on us than what we can do. So what part do you think that we with type 1 can play in making sure that new therapies and technologies are available globally, regardless of socioeconomic factors? I'll maybe mix up and start with Vicky then this time.
That is a really important but difficult question to answer. So I do reflect on this because I am developing a therapy that is based on stem cells that requires incredibly high-end technology and infrastructure to be able to deliver. And clearly the first place where these trials are already happening is not going to be in lower income countries. So I'm permanently aware of that. I think related to that, we are probably not thinking well enough about how we're going to finance these therapies when they do become available. We're no longer talking about a drug that you dole out that costs £10 a week to the NHS. You're talking about a treatment which has got a massive upfront cost, but is life-changing and changes people's future health economic burdens. So I think economics, we need to be thinking about this at the same time as the science. But in answer to your question, what can we all do as a community? My personal belief is that the greatest intersectionality with economic inequality worldwide is driven by climate change. And I think the way in which climate change is going to worsen inequalities and affect health is a huge subject matter for facing humanity. So thinking about our footprint, our global footprint and our environmental footprint, I think is really incumbent on all of us.
Wow, I wasn't expecting that. Thank you. Sarah?
Yeah, so I agree fully with Vicky. I think in terms of things that the diabetes communities can do, and I'm not a clinician, so I'll say that I'm a basic scientist, so I'm not a clinician, but I would say things like ensuring we're getting good participation in trials as they're coming through for these newer therapies, you know, from different socioeconomic backgrounds, from different ethnicities. I think that's really important, so we know that, you know, these therapies are working in everybody, I think is one area. I think educating people about type 1 diabetes is incredibly important, and I think you as a community are able to do that. I think maybe we can learn lessons from distributing insulin equitably globally, and you know, what are we learning as we're starting to roll those things out, and the rollout of like CGM and insulin pump therapies and hybrid closed loops. You know, how do we get those technologies and medicines into different communities and across in different countries around the world, and can we learn lessons for that can help inform some of these therapies that we're hoping are going to come out from these work. And obviously, as Vicky's kind of reflected on, is a lot of that cost benefit studies that need to be done. You know, how do we show that if you get a new treatment that can prevent someone from getting type 1 diabetes, it's hard to put a figure on how much you save on that person not getting type 1 diabetes and having those subsequent complications or other issues that are costly to both the person themselves but also their family and the health care system. So hopefully that's a few ideas. Yeah.
James, what about you?
With Sarah and Vicky, it's a big issue. It's, you know, if you look at statistics from the IDF, for example, International Diabetes Federation, we know that diabetes has a global health burden, and we know that healthcare expenditure in a country is inversely proportional to like things like mortality. So it's a really, really important subject area. It's not unique to diabetes care. It's, you know, this is across the whole healthcare spectrum. You know, we saw things with the rollout of the COVID-19 vaccines, for example. And that means it's a very big challenge to solve. So what can people do? I think, on top of what's already been said, I think, you know, pushing public policy to try and improve that equitable access to new and emerging treatments and current treatments as well. But this is, you know, it's bigger than any one country. This is a big global issue, and it's not going to be solved easily.
We know. We are worried about it as well, because the therapies that are sort of there right now, they're very, very expensive, and I think we're starting to feel the hope that they're coming, and the worry that we might not get actually access them. So they're really useful. Last question then. What's the best thing about working on the Grand Challenge? And Sarah, I'll start with you then on this one.
I'm really glad I get to go first on this one. So I would say definitely the collaborations, the connections, and the people that we're kind of interacting with. You know, it brings such a buzz to the research we're doing. There's a real excitement in everybody I speak to across the UK that have kind of looped in on this. It's the opportunity to bring on a really fabulous team of young researchers. So I'm going to introduce you to my team in my talk in a little while. And it's also the opportunity to interact directly with people like yourselves living with type 1 diabetes, hearing about your stories, what's important to you. It's always an incredibly humbling experience when you go and do any of the kind of like discovery days and the DUK events and things like that. So, you know, that is it for me. It's a wonderful opportunity. It's helped me to develop as a researcher, as a person, and I'm just super excited about where we can take this.
Brilliant. James, then you next.
Similar. I mean, it's really exciting to be able to bring together a team of bright, motivated researchers with a real mission-driven project and a real sort of clear sense of direction. And I particularly enjoy seeing members of my team, you know, developing as scientists and sort of growing within the project. So that's the most stimulating thing, I think. But above the level of the lab, I think the uptick in collaboration has been huge under the Grand Challenge. And not just, you know, actually forming new projects together, but just the discussion, the exchange of ideas and information has been really, you know, really great to be part of. And we're only just getting going. You know, we're the first projects funded, and we're not even a year in. So this is early days in this process, but so far it's been very stimulating to be part of this sort of growing network.
Thank you. And Vicky?
Yeah, I echo everything. The network, the hope. I can see now well beyond the 5 years of this current project. We've got a lifetime ahead of us now to be working on this and towards this. And also actually really, really good fun seeing people who didn't know what diabetes was now suddenly becoming interested in it. There's a huge snowball effect.
Do you know, like you said, words that just made me smile, like it's hope and fun and energetic. I think I'm starting to feel about the challenge, but hope is the biggest one, I think, that we lost a little bit of. So thank you so much. I'll just check now and see, have we got any questions that the community want to bring, but I might move on and get into the presentations to give you a bit more time. So if that's okay, Tom. So I think we're going to bring James up with his presentation. And so Dr. James Cantley is from the University of Dundee. He's a diabetes researcher and one of the senior research fellows for the Type 1 Diabetes Grand Challenge, specializing in the beta cells and insulin producing cells that are the insulin producing cells in the pancreas. Sorry. His Grand Challenge research project aims to identify, develop, and test new treatments to grow new beta cells and encourage surviving beta cells to replicate directly in the pancreas of people with type 1 diabetes. So if James, are you good to go? I can't see your screen.
You should have my slides. I can see them.
I can see a blank screen just yet.
Yeah, I've got them here. I'll tell you what, I'm going to reshare just to see if that improves things.
We're all good for time, so.
Excellent. Share screen. Okay. There we go. So if you can reload that, please, Tom. Perfect.
Yeah, we're good.
We're in business. Okay, so yeah, first of all, thanks very much to Tom for organising this event. I think it's a fantastic forum you've put together, really important for the type 1 diabetes community. And I think it's great that you're including scientists and researchers in this group as well. So Really pleased to be able to share some of our current work funded by the Type 1 Diabetes Grand Challenge. Okay, so I'm a basic scientist like Sarah. So I've got scientific training, and my research career started in 2002, and I've worked on pancreatic beta cells and the wider islet continuously since that time. Part of the reason is, beta cells are fascinating cells to work on, the cell biology, the physiology of how these cells work. But most importantly, they play a critical role in regulating blood glucose levels, and failure of beta cells occurs in both type 1 and type 2 diabetes. So there's a real ethical and scientific justification for working on these cell types. So in my lab, which I've shown here, pictured in their native environment, up at 9 Miles in Dundee, we work on a number of projects related to the pancreatic beta cell. So we're asking really 3 questions: how the beta cells work, so the kind of nuts and bolts that underpin normal beta cell function. We also work on the alpha cell next door as well. Second question we ask is, why do beta cells fail during type 1 diabetes? So we're looking at some of the early inflammatory responses in beta cells. And third, we ask, how can we restore beta cell function during type 1 diabetes? And that's what I'm going to talk about mainly today. But I think within the lab, it's important to have projects in different areas because they kind of overlap and they cross-fertilize with ideas and insights, and also techniques. So I think this sort of cluster of projects around the beta cells are really sort of fertile scientific environment. So the beta cells, obviously, they produce insulin, and they are embedded within the pancreas in these structures called islets, like little islands of endocrine cells. There are other cell types that are important, like alpha cells, which produce glucagon, which helps to raise blood glucose when your blood sugars drop too low. But the beta cells will release insulin, and this acts to lower blood sugar levels, principally through 2 mechanisms. It will stimulate the uptake of glucose into muscle, and it will stop the liver from producing new glucose molecules. And the net effect is to lower your blood sugar levels. I'm going to make a couple of important points here about the beta cell, because it's not just about glucose-stimulated insulin secretion, because beta cells also sense other nutrients. So the breakdown products of proteins and fats, for example, they sense different hormones from the gut, from the fat tissue, and they really coordinate all of these different signals and release the right amount of insulin at the right time and in the right place into the circulation that flows to the liver. Which is really the ultimate closed loop system, and it does all of this in the background automatically. And of course, we know in type 1 diabetes, we have a profound autoimmune destruction of beta cells. So when people present clinically with type 1, they're in this stage 3 phase, shown on the right-hand side, where the mass of beta cells in the pancreas has been heavily depleted, and there's just not enough insulin to control blood glucose levels. But of course, we know the condition starts much earlier than this. And it's very exciting to see approaches such as some of the immunotherapies like teplizumab coming to fruition, which can potentially prevent people moving from stage 1 towards stage 3. Okay, so we've recently celebrated 100 years of insulin, which is a remarkable life-saving treatment for type 1 diabetes. It's effectively turned the condition from a death sentence to a lifelong manageable condition. But I'm sure as many people on the stream tonight will know, there are limitations and caveats to its use. Difficulty matching supply and demand, the inconvenience of this, problems with weight gain, with hypoglycemia, etc. And part of the problem is the insulin is being delivered sort of peripherally, just under the skin. And you need to do quite a bit to get the levels in the liver to a point where you can suppress glucose production. I should caveat as well that I'm not a clinician, so I'm not giving any kind of clinical advice here. This is my perspective as a scientist. Now, of course, we know that with hybrid closed loop, we have a much better, more refined system for delivering insulin on demand, but you still have a ceiling there. So the infusion is still out of position. and it's still a glucose-driven system. So it's not fully recapitulating the role played by the pancreas. Of course, a small number of people are eligible for islet transplantation, and this can be very good for people with problems of hypoglycemia, for example. But there are challenges with donor supply, very limited numbers, the need to suppress the immune system, and poor long-term function of these grafts. And then finally, the very, very exciting field, the stem cell-derived cell transplantation approaches. Some of these differentiate the cells to beta cells, some to an earlier stage cell type, but there are multiple labs and several companies working in this space. And we saw some really exciting data from the ViaCyte device. They reported a few years back. So we now have proof of concept data in people that these transplants can produce insulin. Having said that, there's still some pretty big challenges to go. Managing the immune system, the regulatory pathway, the cost that's going to be associated with the cell therapy that may limit wide access to this technology. And also, we don't really know how durable these grafts are in the long term. But a very, very exciting approach, and I guess relatively close to clinical application. So what we wanted to do is we've with this project, we sort of stood back and looked at the current landscape of diabetes treatments. And what was clear to me is that you've got things that are available now, things that are close to being available. But what if we take a longer view here and we say, okay, what if the ultimate goal for treating type 1 diabetes is to restore beta cell function in the pancreas, to give some of these their own beta cells back, if you like? It's an ambitious goal, but how can we put the first steps in to try and de-risk this approach, to sort of get some momentum and to start to progress this towards potential future clinical application? And after lots and lots of thought and planning, I really settled on two pathways to focus on for this fellowship application. One is seeing if we can stimulate formation of new beta cells. That's called beta cell neogenesis. And the second, can we stimulate beta cells that are still there, that have escaped the immune system and they're still in the pancreas? Can we encourage these to duplicate, and those cells to duplicate, to kind of regenerate a functional beta cell mass? Now, everything I'm going to show now and I'm going to talk about is at the preclinical stage. So we're in the lab working with cells and models. This is not in people. We hope that will come after the 5-year project. But we're doing what we call the preclinical phase, where we're trying to develop and test these ideas and these approaches under this Grand Challenge project. Okay, so the first arm, neogenesis. So during development, that's embryonic development, when the embryo's in the womb, there's very, very high levels of beta cell formation, this process that's kind of summed up as neogenesis. And there are specialist cell types within this kind of branching, developing pancreas that form the endocrine cells, as you can see in this very nice animation here from Eurostem And whilst this process is very active in development, we know that in the adult, it essentially switches off. It's extremely low levels of activity. You occasionally see some neogenic activity, but it's very, very low. It's essentially switched off. But we know if the pancreas is injured, this neogenic program can switch on very, very quickly within a matter of days. And we see this both in rodents and people. So this encouraged us to think that, okay, is there a way we can find a molecule, like a drug, that can stimulate reactivation of this neogenic pathway? Because all the genes coordinating this process are still there. They're just switched off. So can we restart this program in the adult pancreas for a short period to stimulate formation of new beta cells? And effectively, what this would do is to put the right cell back in the right place in somebody living with type 1 diabetes. So that's really the rationale and the vision for the project. So how are we going to do this? So I'm going to keep this very brief, but effectively what we've done is to design what's called a phenotypic drug screen. So phenotypic means we're trying to model a process that happens in the body in a lab test, something we call an assay. And we've put a lot of time and effort into digesting it away the pancreas and purifying our target cells. And we've learned how to keep these happy in culture. And what we mean by in culture is to sort of trick the cells to thinking they're still in the body. So we control the temperature, the gases they're exposed to, the nutrients in the media, all of these things to try and keep the cells happy as if they were in their native tissue. And then, of course, we can treat these cells with drug candidates and to see if they switch on beta cell genes. So this is obviously a large, complex project. There are lots of people involved, lots of different labs involved. And I'm going to show you how this kind of fits together as part of the project. So we have an assay preparation phase, and then we go into our drug screening program, where we can select different molecules from a vast chemical space and test these on our assay. We can then validate these, very importantly, in different systems in my lab. And then we can go on into a phase of what I've called here drug development. So effectively, the chemistry team can sort of tweak the molecules, and the biology team can test how well these work, and we can start to improve any hits we get from our screen. It's also very important we understand how these putative drugs work. So we'll be going on to look at mode of action, finding out what proteins they interact with in the cell, looking at how they regulate gene expression and what these genes do. And really, I think there's kind of two outputs from this. You know, the most optimistic output is we end up with a drug candidate that can go forward to preclinical evaluation. That's the best outcome. But the worst outcome is that we don't get that, but we do generate a lot of new insights into how beta cells form and how they behave. And that's really important for the wider type 1 diabetes research community, because that could be useful for informing, say, stem cell differentiation programs or other aspects of research. So I think really we've got a kind of win-win situation with this project. I should also caveat that this is early stage drug discovery, which is inherently high risk. So the chances of finding a new drug are small, but we are giving ourselves the best possible chance of doing that with this program. And of course, the hits that we've developed will go on and undergo preclinical evaluation. And we primarily do this in a mouse model of type 1 diabetes. And if this is successful, we can then look to move forward towards a future clinical stage. Okay, so the second arm of the project is targeting this beta cell proliferation. And you can see here in this schematic, the idea is that you can give a drug that can encourage a beta cell to duplicate and those daughter cells to duplicate. But the one problem we have is that beta cells don't like to duplicate, and they're very committed to their cell identity. They're highly differentiated is the sort of scientific term. But there's quite a rich literature showing that using different genetic or pharmacological methods, you can actually encourage them to proliferate. And so I can't say too much about this program in this sort of public forum, but I will say that we're working hard to test strategies to drive beta cell proliferation in a preclinical context. And the idea is that we can increase the beta cell mass during type 1 diabetes. So finally, I'd like to just mention we need to improve our tools as well. So conventionally, on the left, we would measure beta cell mass by sectioning the pancreas at 4 discrete levels. As you can probably imagine, we're only looking at a tiny amount of the pancreas with these 4 little sort of slices through the pancreas. So what we've done is to come up with a new reporter strategy where we can express a fluorescent protein. It's called TD tomato. As you can probably guess, it's red. And we can express this under the control of the insulin gene. So basically, our beta cells will glow red. And combined with a clever way to clear the tissue and a giant microscope that's available from one of my collaborators, Gail McConnell, we can now image all of the beta cell mass in the entire intact 3-dimensional pancreas, which gives us a lot more information to work with when we're evaluating drug candidates as part of this project. And I've got a little video here just showing us sort of flying down from the top to the bottom through the entire pancreas. And you can see the little balls of beta cells, the islets sort of flash past as we go. So this is a really nice technique now, so we can capture that entire beta cell mass with all its sort of anatomy and context there. Okay, I'd like to also emphasize the sort of where this project fits in the kind of therapeutic timeline. Often when we give talks, we talk about our area and the technology that we're most excited about, but I really want to emphasize that there are really exciting things happening now with improved diabetes screening, with immunotherapies, and a good chance of preventing progression if you can find people at a pre-diabetic phase. There's really exciting things happening with stem cell derived cell transplantation, and this is real, strong midterm potential. But I think it's really important from a scientific perspective, and also from the perspective of people living with type 1 diabetes, that we have these longer range, higher risk approaches. So can we put the groundwork in now that could potentially lead to a new approach or a new clinical approach in 5 to 10 to 15 years time? And that's really where our project sits. And that's been fantastic to have the Grand Challenge supporting this type of research. I'd like to introduce the people involved. I'll start with the core regeneration team in my lab. So along with myself, we have Lisa Logie, who's a very, very experienced postdoc, both in biotech and academia. Morgan Shaw, who's a research technician who's got a lot of experience with human pancreatic tissue. And Greg Jackson, who's a PhD student in my group. And he's specialised in microscopy and gene expression studies. We also have been able to put together a really excellent collaborative network, so 9 international collaborators, biggest team I've worked in, and it's been really only been made possible by the Grand Challenge funding. And then, of course, the other fellows. We interact a lot, we talk a lot, and this is a really fantastic developing network. I should really update this slide because there are now more projects funded across the UK and a real expanding Grand Challenge network. And finally, just to summarise, we have, I think, a very exciting early stage drug discovery project. We have 2 shots on goal with the neogenesis and the proliferation arms, but it is a high risk, high reward game that we're playing here. And some reflections that I've made throughout this process, I think funding this type of research is extremely important. I think the diversity of projects in the Grand Challenge is also very important, that there's different aspects of science being receiving funding. And as the point just came up in the discussion earlier, the ability to build collaborative networks and play as part of these big teams is really, really important. And finally, the involvement of people living with type 1 diabetes. So I've been very privileged to sit on a clinical studies group at Diabetes UK for a number of years, and hearing firsthand the sort of challenges that people face. And also, I'm very grateful for the time people spend reviewing the plans for our grant applications and things like that. So it just remains me to return to my lab and thank everybody here and also our funders over the years that have funded our research, and just to leave you with a picture of Dundee, where we're based in northeastern Scotland. So back over to you, Mary.
Yeah, thanks, James. We've got a couple of questions in the comments. So, James, so we can look to activate beta cell neogenesis as a good objective, but presumably they would still be vulnerable in an immune attack in type 1. How will that be overcome?
Excellent question, Steve. That's a really important point. I didn't have time to go into that given the time here, but we now have an FDA-approved immunotherapy, teplizumab, effectively the first generation in clinical use for prevention of type 1 diabetes. And of course, there'll be new generations of those immunotherapies available. So we have a tool that's approved for use in diabetes prevention that could potentially be applied to protect regenerated beta cells. So that's a real game changer for the regeneration field when teplizumab became FDA approved. And we'll be testing that in our preclinical work as well.
Brilliant. So this is quite a long one, let's see. Maybe it's just a comment replying to that. We met at the Diabetes Scotland volunteering event in November. The information from James and the passion from his team, a few with personal links to type 1, really came across. Can't wait to see results from Dundee and the other Grand Challenge centres. So that's a comment from.
Thanks for the comment, and I really enjoyed attending that event. And all of my team came along and got up and spoke as well. So it was a great event. Looking forward to the next one.
Stephen Nixon, yeah. So okay. I think that's us bang on time actually. So we'll move on then to Sarah's presentation. Thank you so much, James. And so next up, we've Professor Sarah Richardson from the University of Exeter. Professor Richardson co-leads the islet biology team at the University of Exeter Medical School. She's been researching diabetes since 2007. Her project will investigate how and why the immune system destroys insulin-producing beta cells in type 1 diabetes and how the process may differ between people with the condition. This is one I'm really interested in myself because we've got two type 1s here in my house, but they seem very different. So very
Thanks, Mary.
interesting here. Thank you.
Thank you, Mary. Okay, brilliant. So we've got some slides up on the screen, which is a good start, and I'm feeling the pressure to stay to time. So I'm, without further ado, I will get moving. Those slides moving forward, okay. Yep. Okay. Basic. So thank you very much for the invitation to come and talk to you today. I'm really looking forward to hearing some questions from you about the work that we're doing. And I'm going to be telling you today about how we're leveraging new knowledge from the human pancreas to try and advance and improve our understanding and our treatment of type 1 diabetes. So what I want to do, first of all, is introduce you to my fabulous team of researchers. This is a team based in Exeter. We have Christiana, Katie, and Ty, who are postdocs within the team, and Steph Hunter, who is a PhD student. And all of these individuals have been working tirelessly for the last year, trying to develop some of the ideas that we've put forward into our a Grand Challenge. We also have Sylvie, who will be joining us in October, who lives with type 1 diabetes. And I'm really looking forward to having Sylvie on the team as well, as she'll start to give us a really unique perspective into type 1 diabetes and have someone on the ground all the time giving us some input there, which is fabulous. I also want to acknowledge the Islet Biology Exeter team, and this a great community of young investigators, all looking at slightly different aspects of type 1 diabetes research that complement one another brilliantly. So bringing lots of different expertise within Exeter. And of course, I want to really acknowledge Professor Noel Morgan, who's been a fabulous mentor to me over the years. And I'm very lucky to be embedded within the EXCEED Centre in type 1 diabetes, with the Exeter Centre of Excellence in Diabetes Research. And as we've kind of already touched on, during our conversations that we've had so far, I really want to illustrate kind of what the Grand Challenge has done for UK type 1 diabetes team science. So I've told you a little about the team in Exeter. I'm really surrounded by this great team of basic clinical and data scientists who are working in type 1 diabetes, who are really helping to support this fellowship role goals. But there's also a really strong collaborative network with other type 1 diabetes researchers from right across the UK. And those I've highlighted in red here are collaborators on this fellowship. But even, beyond that, we also have access to a much wider network of researchers through the UK Type 1 Diabetes Research Consortium. And in green, of course, I'm going to highlight James Cantley and Victoria Salem, the other two SMF fellowships holders who we've been hearing about and from today, and really to emphasize that our programs are designed to complement one another and really fit with the wider Grand Challenge goals and projects that are now emerging in the later stages of this program. So really can't emphasize enough what this Grand Challenge has done for type 1 research in the UK. So going back and sort of talking a little bit about the type 1 diabetes timeline here, we know that we have individuals who are at genetic risk of developing type 1 diabetes. And in those individuals, they experience at some point in their life a trigger, which could be, for example, an enteroviral infection that sets off this process of autoimmunity. And this can be picked up in individuals who don't yet have type 1 diabetes by looking in the blood for either islet-specific autoantibodies or islet-specific immune cells. And once those things are present, we know that we can that there's this progressive beta cell destruction and dysfunction that eventually leads to the clinical onset of type 1 diabetes that you're all very aware of. But essentially what's happening in this kind of timeline here is a little bit of a black box, because in order for us to be able to understand that, we need access to the pancreas, which is the site at which this disease is occurring. So what makes the work possible that I do is availability of human pancreas for research. And sadly, this is a really precious and rare resource, and there are actually less than 700 type 1 diabetes pancreas reported worldwide, and less than 400 of those are available for research. And so that's the type 1 donors, but we even have restrictions in the amount of pancreas that comes from individuals without diabetes. And in order for us to understand the disease process, we have to understand normal first. So it's as important to study pancreas from individuals without diabetes as it is individuals with type 1 diabetes. And I'm incredibly lucky in Exeter to host and curate a very unique biobank of tissue that comes from individuals who've sadly died close to onset of type 1 diabetes. Because as you can see here, and I'm sure you're all aware, the pancreas is buried very deeply in the abdominal cavity. It's very difficult to access, and you can't biopsy it like you can other organs because there's a big issue with potential complications from that surgery. So sadly, the only pancreas that's available for research comes from individuals who sadly died. So these rare pancreas bioresources are incredibly precious. And we have one called the Exeter Archival Diabetes Biobank in Exeter. But through the various collaborations that my team and I have around the world, we also access a wide range of other biobanks to ensure that we can look at as many pancreas samples as possible. And as such, we've probably examined more type 1 pancreas than anyone else in the world. So I think in order to give you some perspectives on that, I want to show you a little bit about what a typical pancreas section might look like. So once we collect a pancreas, that pancreas is sectioned and put into these paraffin wax blocks, and then we cut very, very thin sections of the pancreas onto slides, and then glass slides, and then we can stain those pancreas sections to help us to visualize the cells in there. And so this is what a typical pancreas section might look like in an individual without diabetes. So essentially you can see this kind of sea of blue, and this is the acina part of the pancreas, and this makes these cells in this part make all your digestive enzymes that help you to break down your food. And then you can see these kind of islets, or the islets of Langerhans, and these are the endocrine part of the pancreas, and is a part of the pancreas that produces all of your hormones that help you to regulate your blood sugar. And it kind of, when you look at it like this, it almost looks like those little islands are kind of in a sea of dangerous tissue, and that's kind of how I've likened it here in this picture here. So it's almost like these little islands are floating in this rather treacherous landscape with this acina tissue that can produce digestive enzymes that could damage the endocrine cells themselves. And that really isn't too far from the truth. So the healthy islet is a little bit like an island fort. So they actually have defensive walls around the outside of the islet themselves, and that's called this peri-islet basement membrane that you can see around the outside of this islet that's shown here in blue. And I'd like to thank Mike Miles Boardman, who's an illustrator who lives with type 1 diabetes, who helped me put these slides together to demonstrate what the pancreas looks like. We also know that the healthy islet has a very diverse community within it. So we've got that wall around the outside, and we've got these different types of cells, all of which work together to help with specific roles to help to regulate your blood glucose levels and ensure that they're controlled appropriately. And finally, the islets also have a transportation hub, or the islet vasculature, which allows fuel and your oxygen to come into the islet. So for example, your glucose will come in here, the cells will communicate with one another, respond to that, and then secrete insulin, and all the waste products will come out through these little capillary or smaller vasculature that comes out from the islet, and that will then be transported around the body as required. So what do islets look like when we look at an individual with type 1 diabetes? So this is a pancreas section from an individual with short duration disease. And already I hope you can see some differences where all the islets in the last pancreas tissue that we looked at had slightly pink looking to them, which is where we can see the beta cells. You can see here that there are big regions of the pancreas where there's just blue islets present, and this represents islets where the beta cells have already been destroyed. In other areas, we can see islets that look relatively normal with lots of pink cells present. But if you look down here in this lobe at the bottom, hopefully you can see these small brown dots around some of these islets, and those are the immune cells. So in these short duration individuals, we can actually see the active immune destruction of beta cells happening. And when we study those, and it's almost like that islet is under siege, we can find islets across those sections that are at different stages of that immune mediated destruction. So we can see, for example, evidence of early scouting parties around the outside of those islet walls that you can see here around the outside, and we call that mild peri-insulitis. As those forces start to mass around that wall, we can see that around this wall here. They haven't yet managed to break that wall down, but when you get the full invasion and they've managed to break through the islet basement membrane, we can actually see these immune cells actually getting into the islets. And then that's at the point where the beta cells are destroyed. And we see that there's complete destruction of the beta cells. All those pink insulin producing cells in this islet have been destroyed. And we're lucky with these pancreas sections then to take these types of tissue sections, and we can then identify all of the different types of islets within a given pancreas section and start to plot that ongoing disease process. So what that helps us to do is to identify which immune cells are present at the different stages of destruction, and that is going to help guide the immunotherapies that we've been hearing a little bit about already, and help us to identify the ones that are going to be most effective at slowing or preventing that loss of the insulin producing beta cells. So sort of summing up, what my team essentially is doing is taking these unique pancreas biobank resources. We're doing very targeted staining and scanning. We can visualize those slides in different ways and develop pipelines that enable us to determine what's happening within the pancreas at different stages of the disease. And my fellowship essentially is then broken into two work packages. One, looking at root causes, so what might be triggering type 1 diabetes and how does it develop in different people at different times. And then we're also contributing to looking at strategies that could help to enhance islet defenses. And finally, we work with researchers around the world to use it as a translational resource to ensure that their studies they're doing in the lab might actually translate into therapies in the future. So really, as I've mentioned before, to fully understand disease, we must understand normal. We've got very limited knowledge of human pancreas development in young individuals under the age of 10, and even more limited understanding of what's happening in pancreas close to onset, particularly in young individuals. So we're really taking these careful observations of these rare pancreas from individuals with and without type 1 diabetes to develop a better understanding how beta cells could be protective and what's going to be the most effective way to refine therapeutic targeting. And really, we hope that we're going to strengthen our understanding of type 1 diabetes, reveal novel therapeutic targets, and inform clinical trial design and patient-specific therapeutics in targeting in the future. So in our first work package, we're going to build on our previous track record in defining root causes, and in particular, focus on work that we have done defining pancreatic type 1 diabetes endotypes. So our unique findings have suggested that there are different forms of type 1 diabetes that are potentially driven by different underlying disease mechanisms. And encouragingly, these observations from the pancreas align extremely well with clinical observations where we know that not not everybody's diabetes is the same. So briefly, if you've got type 1 or type 1 diabetes endotype 1 or type 1 individuals, we know that those individuals tend to be diagnosed and they have evidence of a more aggressive immune response with very few beta cells remaining at onset. Whereas in contrast, individuals with TIDE2, they tend to be diagnosed older and retain their beta cells for many years following diagnosis. So in the first kind of work package, we're aiming to address why these pancreatic endotypes might exist. So we know that the pancreas grows really dramatically in early life. So here we've got the age of individuals and the pancreas weight. This is just in individuals without diabetes. And you can see that there's a really dramatic increase in the size of the pancreas in the first sort of 10 years of life. And at the time where we think we see the first signs of autoimmunity appearing in different pancreatic endotypes, there is this massive 4.5-fold increase in pancreas weight. So essentially what we're trying to do initially is use individuals, a pancreas from individuals without diabetes at different ages to try and plot what and define what's happening to the pancreas, how it's changing and restructuring during early life, which might give us clues as to why the diabetes looks very different in these two different groups. So we do that by mapping the pancreas restructuring during early life. So we take these different scans of tissue and we use something, some AI-based image analysis software called Halo to help us analyze the pancreas sections that have been stained with markers that allow us to look at different pancreas tissue compartments. And then we train the AI to recognize each tissue region of interest, and then we can analyze the whole pancreas tissue from individuals both with and without diabetes across different ages, and we can export all that data in relation to the different pancreas types, such as the acina, which I've been telling you about, duct, and of course importantly the endocrine part, the part that produces the hormones that control your blood sugar. And that helps us to map how the endocrine in particular changes during early life. So here we have a pancreas from a fetal pancreas at 12 weeks post-conception, and the blue dots you can see on here are our islets of Langerhans. This is a donor that's 9 years of age, and you can see that, you know, just from the get-go that's very different, that there's many fewer islets and they're dispersed further apart and they get larger with time. So when we quantify that, we can see that there's this dramatic change and restructuring of the pancreas during that early phase of life. And what we've been able to show is that as you age, we start where there are lots of very single cells, endocrine cells, that form into small clusters, into clustered islets, which then, as the pancreas is going through that big growth, they are separating from one another until we get our larger islets. And when we study the pancreas from individuals with type 1 diabetes and compare that to what's happening in individuals without diabetes, what we find is that these small islets are the ones that are targeted by the immune system in type 1 diabetes. So if an individual gets type 1 diabetes at a very young age, where we're seeing a lot of these very small islets before they've had an opportunity to grow into the larger islets, they may never have the opportunity to develop into these bigger islets. So we've got fewer islets there in the first place, suggesting that stem cell derived beta cells may be a more viable therapy, or that we need to work out individuals who are at risk of developing type 1 diabetes and treat them with some of those immunotherapies that prevent that destruction early on and enable the individuals to grow more of their own islets. We're also, as I mentioned, looking at that islet basement membrane that we found around the outside and trying to study that in more details. We know in the adults they have this islet basement membrane, but we really don't know when that basement membrane develops during early life. And we are hypothesizing that in young individuals under the age of 2, that peri-islet basement membrane may not be fully intact, which means that they're more vulnerable and the immune cells can effectively break through that wall. And you can see that here in the adult, you've got the islet basement membrane around the outside, but in a 2-year-old, you can see that that is not yet fully formed, which means if you have an individual who has got type 1 diabetes and immune cells in the pancreas that can kill the beta cells, they can break through that wall very easily. And we're also working with Chloe Rackham and Rebecca Dewhurst-Trigg, based in Exeter, to look at accessory cells that help to build that islet basement membrane and chart when they appear and develop in those individuals. We'll also then move into individuals with type 1 diabetes and ask in those endotypes, how does that islet basement membrane look in those? And we predict, because we see more of that invasive insulitis where the immune cells have broken through that basement membrane, it's compromised, that we're seeing that sort of process ongoing in those individuals with TIDE 1, whereas in individuals with TIDE 2, that basement membrane appears to be more intact and the immune cells are kept out. So what can we learn about that? How can we potentially engineer basement membrane to protect our islets? In our second work package, we'll explore strategies that islets may be using to defend themselves. So we've previously shown that islets from individuals with type 1 diabetes express molecules like classical HLA that are detrimental, and they make the islets more visible to the immune system, and so could be contributing to beta cell demise. But they also express molecules like non-classical HLAs that you can see here in the green, which are immunoprotective. And we've been determining if these molecules are differentially expressed in the type 1 diabetes endotype, with the hypothesis that the protective molecules may be more prevalent in the TIDE 2 donors who retain their beta cells for longer. And these HLA molecules all require direct cell-to-cell contact to communicate with the immune system. But as I've shown you, we have that basement membrane around the islets that would prevent that direct contact between these molecules and the immune cells. But we know that these molecules can also be generated in soluble or extracellular forms. So we hypothesize that there are soluble forms released from the effective islets, and that these are the factors in dialogue with the immune cells. And we've now been able to confirm that the soluble classical HLA-1 molecules are released from cultured human beta cells, and these are increased following treatments with stressors associated with type 1 diabetes. So you may be asking why this matters. And so really, if we want to protect any remaining beta cell an individual may have, or as we move towards beta cell replacement therapies, there is a scope for us to use these soluble immunomodulators alongside the repair or the regeneration of that basement membrane as protective strategies as we move forward. And then the final work package that we've been working towards is really collaborating with many different groups across the UK and across the world to take the data that they are getting from their beta cell culture models, so things in the lab that you've heard about, like for example from James, looking at people who are working with these stem cell derived islets and isolated human islets and mouse models of diabetes, and each of them will be identifying particular targets for treatment and their models, and we've been trying to translate those into the pancreas tissue to confirm whether those are really good targets for us to go after therapeutically, by seeing if these are aberrant or not working the way they should do in individuals with type 1 diabetes. And we've had a good deal of success in the first year with that. So it just remains for me to thank you for your attention. Particularly, I want to thank the donors and their families, without whom none of this work would be possible, and all the many collaborators across the world who have contributed to the work that we've been doing. So thank you for your attention, and I'm happy to take questions and pass over to Mary.
Thanks so much, Sarah. We're going to move to Vicky next, but there are comments in the comment section. If you can have a look at them, that would be brilliant. And Thomas brought them up. So they're about your slides basically, and your graphics, and they're how wonderful they are. But I'll let you read them in the comments, because
Thank
they
you.
are very
Sorry,
lovely.
I went a little bit over. Apologies.
Very interesting though. Like, I'm actually going to have to listen to it all back again and go even slower, because it really applies to my story here. So thank you for it.
Thank you.
So next up, we have got Dr. Victoria Salem from the Imperial College London. Dr. Victoria Salem is a clinical senior lecturer in diabetes and endocrinology at Imperial. She researches the islets of the pancreas, which is where insulin producing beta cells are and other hormone producing cells are located. The aim of her Grand Challenge project is to develop a device that can be implanted into people with type 1 diabetes to deliver a new supply of beta cells. So while Vicky is loading her slide deck, I can see your slides, Vicky.
Can you see them good?
Yep.
And can you hear me?
Can hear you fine. Yep, go ahead.
So I am
Are you on your first slide though?
No, I'm not. Hold on a minute. Can you see that first slide?
I can't see the first slide. I can see with a blue and a red.
Yeah, hold on a minute. I'm trying to find the right window. Just bear with me.
Sure.
Oh, I see why. Okay.
We're all good.
Sorry about this. Can you see that one?
It's loading. Yes, that's you.
Excellent. I apologise.
Right.
I thought I had that licked, but clearly not. So I can now just see my screen. So do shout out if the slides aren't following. But thank you very, very much indeed for inviting me to be here today. So I'm Vicky Salem. I'm the third of the 3 fellows awarded in the first round of the Grand Challenge funding, and I am a clinician scientist. So I am a diabetologist. I run a type 1 diabetes clinic at St Mary's Hospital in London, but my lab is based in the Department of Bioengineering. In fact, my first degree before medicine was bioengineering, so I feel like I've come home a little bit. And my lab is at Imperial College on South Kensington campus, where I'm working with engineers and basic scientists as well as clinicians towards a cure for type 1 diabetes. So I really like this slide because it reminds us of the mental burden of living with a chronic disease. And I'm sure many people in the audience will identify with this. We know that living with diabetes means hundreds of more decisions and anxieties every day associated with having to measure your own blood sugars and dose your own insulin to basically be in charge of your own metabolism, a huge burden. And of course, we all know about the complications of diabetes, classically microvascular complications, imperfect blood sugar control can damage blood vessels, and the small blood vessels in the eyes and the kidneys and the nerves can get damaged. As we get older, we also have an increased risk of macrovascular disease, but we're getting much better at treating diabetes, particularly in high-income countries, and we'll talk a little bit about closed-loop systems that are available to many patients nowadays. So as we're seeing less, and it's true, we are seeing less vascular complications, we're starting to think more and more about emerging complications of diabetes in people who've lived with type 1 diabetes for many decades. And not least amongst those, as I mentioned, is the psychological burden and the mental burden of managing this disease lifelong. So we're all dreaming of a cure. Has AI or the artificial pancreas solved the problem? And again, many of you will be using this type of technology, and I know this from my patients, but I can also tell from some of the comments that arose as we were setting up this evening from people like Tom, that technology has been a huge game changer, but it doesn't feel like a cure. Of course, what a closed loop system, a modern closed loop system consists of nowadays is the CGM sensor, which lasts, if you're lucky, up to 2 weeks and is constantly monitoring your blood sugar. And then at the other end, the delivery system is an insulin pump that is delivering a slow but sure dose of insulin under the skin. And linking those two and communicating between those two elements, of course, is an algorithm, incredibly powerful computer-driven algorithm based on complex mathematics that is trying to use the glucose data to predict where your blood sugar is going and then tell the pump how much insulin to deliver. And we now have a huge number of these types of technologies available to offer our patients. We've got 3 that we've commissioned at Imperial, which our patients can choose from. They differ somewhat, not just in terms of the maths involved in the algorithm, but of course the hardware and the hardware that people have to attach to themselves, and they're great. This is a recent study which has looked at time and range. So that's how much of the day your blood sugars are within the normal target range. So we actually start to use this metric even more than HbA1c nowadays to see how well people are managing to control their blood sugars. And so the top graph here is for children and the bottom graph here is for adults. And the red line is showing those people when they were using a standard insulin pump, but telling the pump what to do themselves, so controlling the pump delivery of background and boluses of insulin themselves. And then the blue line are those same patients using a closed loop system, and this is one of the open source algorithms that some of you may be using. And you can see they're really, really good, especially at night when there are fewer variables at night like exercise and eating and stress. Actually, these closed loop systems can achieve incredible control. But as I've said, it doesn't feel like a cure. The technology fails. And interestingly, actually, when we look at our real world data, when we start people on these closed loop systems, a good 4 out of 10 people are not actually using them as hybrid closed loop systems at 3 or 6 months. And people stop using them because of lack of trust, because they're not sure about where things are going. They get frustrated with certain elements that they don't feel that they have control of. And as I said, many of you will be aware of many of these frustrations. So the technology is getting better and it's going to continue to get better. And it's, as I said, it's been a game changer. But when people think of a cure for type 1 diabetes, they think of a biological cure. So that is using cells transplanted back into a patient with type 1 diabetes to replace the beta cells that were lost during the development of the disease. And I think these graphs here sum it up very nicely. So these are graphs of somebody's, or 2 patients, in fact, patient 1 at the top and patient 2 at the bottom. Patient 1 at the top, we see before a range of their 24-hour CGM glucose profile. So you can see lots of variability and less than perfect glucose control before. Then they start with their artificial pancreas or their hybrid closed loop system. And you can see huge improvements in terms of the average blood sugar. That's going to translate to a massive reduction in HbA1c and a massive improvement in their long-term risk of complications. But there's still a lot of variability, as I'm sure many of you who struggle with these systems occasionally will appreciate. The bottom panel represents a patient who's undergone cadaveric islet transplantation. So they've received an islet transplant of islets that have been donated by somebody who's died, in the way that somebody might receive a kidney or a heart. And just like any other organ donation, this patient will have to be on very, very strong immunosuppression in order for them to tolerate those transplanted islets and not reject them. But look at what happens here. The blood sugars normalize. This is now a patient who never has to measure their blood sugars again, and who is always normoglycemic. This is as close as we can clinically achieve at the moment to a cure. But I said a person receiving this treatment needs to be very, very profoundly immunosuppressed. the long-term risks for that include cancer and infections. This is not applicable for the vast majority of people with type 1 diabetes. So, of course, attention has turned over the last 10 to 20 years of how we can replace beta cells for the majority of people living with type 1 diabetes. And I've spoken to you about cadaveric donor islets. That is not going to be the answer because of the need for immunosuppression and the fact that one person donating their pancreas after they've died is not enough. We need at least 2, sometimes 3 recurrent donations to get enough islets to actually survive the transplant process when we donate them to a recipient, for them to actually become insulin free. So cadaveric donor islets are not the cure. So where are we looking? Well, there are some groups, particularly groups working in Canada at the moment, that are looking into xenotransplants. So looking into the delivery of islets taken from non-human animal models. And you will have heard recently, actually, of the first patient who sadly didn't survive, but he survived for longer than we expected, of the first patient who's received a kidney from a porcine or a pig that was genetically engineered for the cells to be a little bit more human-like. So some research advances there in the use of xenotransplants, but we're not yet at clinical trials for islets from this type of donor. And that's partly because there's been a huge explosion in the development of what we call stem cell derived beta cell replacement therapies. So these are beta cells that are grown from embryonic stem cells. When a sperm meets an egg, an embryo is formed. Sometimes we use an embryo like that for research and or regenerative medicine purposes. And just as that first cell that is formed when an egg and a sperm come together can form into any part of a human body, they will divide and start to differentiate into cells that form a baby. So we have learned how to take those embryonic stem cells and turn them into mature-ish insulin secreting beta cells. So that journey, and I'll talk a little bit about that, is close to fruition. Now the question is, how are we going to take those stem cell derived beta cells and put them into our patients without the need for systemic immunosuppression, which is too risky? And basically, there have been 3 major ways of thinking about that. At the top here, we've got the concept of microencapsulation. So this is taking groups of those stem cell derived beta cells and surrounding them, coating each of these little pseudo islets with a material that is designed to ward off the immune attack, and then injecting these little islets into the liver, which is what we would do with cadaveric islets. Unfortunately, attempts like that have largely failed, and that's because this concept of microencapsulation is simply not strong enough to ward off the strong rejection of foreign cells and ultimately the potential for autoimmune reattack of the beta cells, which of course is what caused diabetes in the first place. There's been slightly more success with the use of macroencapsulation devices. So these are larger devices which tend to have a solid outer surface that is designed to actually ward off the immune system. And these are macroencapsulation devices which can actually be inserted into the patient under the skin or inside the abdomen. And they are, I'll talk a little bit about those later, but on the whole, the risk with these is trying to get the balance of getting enough blood supply into these islets and to avoid scarring around what is actually quite a solid box containing these very, very sensitive and precious stem cell derived beta cells. So latterly, and this is what my lab is interested in, people are thinking about coating these stem cell derived beta cells in much more complex jelly-like materials, what we call hydrogels, which are softer. They feel like human tissue. They're much more biocompatible. They don't exert a sort of inflammatory response. And they also have the added benefit of being very pliable. So we can start to formulate them and the cells that are being carried within them into devices that actually feel a little bit more like a replacement pancreas. And so that's really the focus of the research that we're doing in my lab. So here is my Grand Challenge slide. And I just wanted to give a shout out at this point to my fantastic collaborators in Birmingham and Oxford, who I'll introduce you to shortly. So Jim Johnson is one of the world's leading islet biologists with a strong interest in beta cell replacement therapies, and I hope he doesn't mind. I have nicked one of his slides, but I've edited it a little bit, and this is a slide that is reminding us of the journey that we've taken so far in science, from trying to understand how we might push an embryonic stem cell towards becoming a pancreatic beta cell, and took a number of huge advances in science. And I would say that we're about here at the moment. So actually, we've done most of the hard work. We have worked out how to create a beta cell that can sense glucose and release insulin. But there's still a huge amount of work still to do to get to the top of the summit. So whilst we've got glucose responsive insulin secreting stem cell derived beta cells, they're not as efficient as fully mature adult beta cells. We don't know if they're going to last as long. And so you need more of them for a given number of cadaveric islets to treat diabetes. So still a huge amount of work to do to get to the final stages of the development of a fully mature, fully functional insulin secretory cell from a stem cell. Nevertheless, we are now doing this routinely in the United Kingdom, thanks to Diabetes UK and latterly Grand Challenge funding. This is Ilda Ackerman, a fantastic scientist, a geneticist by training, who set up Beta Cell Birmingham, the first centre in the United Kingdom that is routinely making stem cell derived beta cells from human embryonic stem cells. She uses huge bioreactors which are constantly spinning, and she has to go in at exactly the right time of day with exactly the right cocktail of different growth factors and hormones and chemicals that are gently trying to push each of these stem cells through the natural developmental milestones that occur in a developing human fetus. So we've used the fetus and the development of stem cells through to beta cells that occurs during fetal development. We've used that as inspiration for how we create our mature stem cell derived beta cell clusters from embryonic stem cells. This is, as I said, hugely painstaking work, which is half science and half art. But as a concept, the game changed big time a couple of years ago when Vertex, which is the biggest biotech company, a pharma company working in this field, announced its clinical trials of their stem cell derived beta cells delivered to patients with type 1 diabetes. And again, I have copied this data from one of their slides that they presented at the EASD conference at the end of this year. So Vertex are now delivering stem cell derived beta cells into people with type 1 diabetes. They have to be immunosuppressed because these are embryonic stem cells that are coming obviously from a donor that is not matched to them. So they have to, just like a cadaveric transplant, they need to be immunosuppressed. But look, we now have at least 3 people who have received stem cell derived beta cells for well over a year, in some cases over 2 years, who have no longer got diabetes. In essence, they are normoglycemic for the vast majority of the time. So now, the game has changed. The concept of being able to use stem cell derived beta cells clinically is here. But why are we not yet able to offer these cells to the vast majority of people living with type 1 diabetes? And it's because largely we still haven't cracked the problem of being able to get enough of these cells in to survive for a long time because they've got the right blood supply, and crucially to avoid them being rejected by the recipient's immune system. And so I'm really focusing on this side of the climb to the apex, which is the development of we think, hydrogel-based products that will help us overcome those barriers. I did mention that I'll talk a little bit about some of the attempts to do macroencapsulation devices. So remember, I told you that the early attempts were to take those stem cells and to put them in a hard capsule, which then gets inserted under the skin of the patient. This is the ViaCyte product at the top. The Beta Air system is a sort of earlier version of that, where you had that hard capsule inside the abdomen, and those two ports that actually had to be exteriorized, had to be sitting outside the patient's tummy for the patient to be able to actually attach a syringe to pump it up with oxygen to keep the cells alive. And at the bottom, this is a Sonova cell patch. There's the pouch there, that sort of nylon-like sheet, which is inserted under the skin in an attempt to vascularize at first. Again, these have failed very sadly in the first run of clinical trials because they get fibrosed, they get scar tissue. And actually, when you go back in to try to fill up those rods with islets or stem cell-derived beta cells, it's almost impossible to get them in. So these hard macroencapsulation devices at the moment don't look like they're going to be the answer. So we're working, as I said, on these two elephants in the room. How are we going to get these stem cells into patients and overcome the barriers of insufficient vascularization, which is hugely important to keep them healthy and happy, and immunoprotection? And I'm just going to cover the immunoprotection bit again with somebody else's work. Doug Melton, many of you will have heard of, is the grandfather of stem cell science. He is the father himself of children with type 1 diabetes. He devoted his career decades ago to trying to find a stem cell replacement therapy for type 1 diabetes, and he really has led the field for all of that time. He's now looking towards the genetic editing of these stem cell derived beta cells. So CRISPR editing them to actually remove the proteins that are expressed from those cells, which make them recognizable by the immune system, in a sense, making them immune cloaked, so that we don't have to worry about the recipient rejecting them. And I think this is where a lot of the biotech companies are going, and that's a hugely exciting area to keep an eye on moving forward. But as I said, the other big area to concentrate on is how we get these cells into the body and how we get them to vascularize as well as being immune protected. And so I'll show you some of the data that we have started to collect towards that goal. We're interested in bioprinting. So this is a schematic of a bioprint, and this is work that I'm doing with Professor Molly Stevens, who's the lead for the UK Regenerative Medicine Network, an example of someone who through the Grand Challenge is really focusing her huge expertise towards type 1 diabetes. This is the concept of a bioprinter. It's a bit like a tube of toothpaste that is extruding our material, and our material is cells, so a mixture of the cells that we want to work with, including the stem cell derived beta cells that are encoded or encapsulated in this jelly-like material, which we can control chemically, and we can print into very complex shapes that start to resemble something more like human tissue. And so our concept is to use complex multi-composite materials of different cells and different gels, which are designed to do different functions within the 3D printed construct that we're making. So here you've got a schematic where you've got these blue columns here representing gel-coated stem cell derived beta cell clusters, and the gels that we're developing to surround these both have an immunomodulatory function. And I'm using a lot of the work that is coming out of Sarah's lab, trying to identify what are the important protective elements of the basement membrane to help us design those materials. And at the same time, we're also co-printing other materials which are really focusing on the vascularization. Getting these cells well vascularized and well implanted is absolutely key to their survival and their durability. I'm really proud of this slide. This is an example of a blood vessel network that we have grown outside of the human body from cells that have been taken from patients with type 1 diabetes. So these are autologous cells. We take the cells from a blood sample, we grow them up in the lab, and then we can actually, you know, print them with our hydrogels, and we can see them grow into these new blood vessel networks. We think this is hugely important for understanding how we can make these devices that can then be transplanted back into the patient. We can also, I don't know if you can see this video, we can actually perfuse real blood through these blood vessel networks, and we can perfuse them around islets. And those islets are very happy once they receive that blood supply. In fact, they're happier than if they were just sitting in a culture medium on their own. And I just quickly put this slide in. I wasn't going to put it in, but this is an example of islets that we're starting to coat with some of those basement membrane elements that Sarah is working on, and we've got some preliminary data to show that when we coat our stem cell derived beta cell clusters, they function much better within our blood vessel networks. And finally, this is the last video that I'll show you because I really love this. It's literally a hot off the press. I don't know if you can see the video playing. You won't be able to see it because I can't press play, but this is a blood vessel network that is grown from a patient with type 1 diabetes. It's glowing green because we've made it fluorescent. You can't see it, but in between that blood vessel network, we've got islets, and those little blue cells which are running through the blood vessel network there. And this is all grown outside the human body from single cells from scratch. Those blue cells there are actually T cells taken from that same patient with type 1 diabetes, which are actually searching out the islets in between that network. So now, as well as engineering cures for type 1 diabetes, we're also engineering platforms that can help us directly study the disease process for the first time in human tissue. I'm going to finish it there. It was a real whistle-stops tour, but thank you very, very much to all of my collaborators, and particularly to my fantastic patients who are a never-ending source of inspiration and love. Thank you.
Oh, thank you so much, Vicky and Mary. Your microphone was on mute there. I'll hand back to you. Wow, that was I think that was absolutely fantastic. So
it's the build of the story.
The
build of how hard actually to research like the pancreas is. I don't think we really understand how difficult it has been, and but to finish it with your now able to engineer something that you can use for diabetes research too in your lab, Vicky, is just astonishing. Thank you.
Thank you so much, Vicky. What a fantastic night, Mary, hasn't it been? And I've got a few comments I wanted to share before we leave. We didn't plan this, but it's just brilliant to see the community really tuned in tonight. We've been on different social medias. We've been on our website tonight. Just under 500 people have tuned in over the course of the night, which is absolutely fantastic. And I wanted to share some comments from Anna. She says, as a diabetic since 1960s, this discussion is flipping amazing. I'm currently being considered for islet cell implant here in Canada, and hoping my participation will help future type 1 diabetes lives. Wow.
Amazing.
Wow. Isn't that just great, isn't it? Obviously, Anna again, she said Sarah's slides with the artwork. I think all three presentations tonight have been absolutely amazing, and
really accessible.
Yeah, understandable to people just like me and you, Mary, living with diabetes. We've had parents and carers listening on tonight, and it's all accessible, understandable. And I think the slides really just made it even a more special evening tonight, because it brought it to life. We've done 5 research events, and this is our second now we've done over this sort of avenue rather than audio. And I think bringing the slides on and the animations by by all three presentations. It was really great. Again, JDRF posted, thank you, Sarah, for giving us a tour of the pancreas. That was just so interesting to see, wasn't it? Like, I honestly didn't know that. Like, I've taken away so much from tonight. Absolutely. And also, Mary, you put this as well. I don't know if you wanted to elaborate on
Yeah,
it.
no, I did. I learned so much from Sarah about Johnny. Johnny was diagnosed when he was 6, and I was diagnosed when I was 18, and how I felt when my diabetes was to how I experienced his were very different. And just seeing that in the Type 1 and Type 2, and it's just fascinating. Yeah. What can we do? Like, can we do anything to help? Because we know diabetes research and accessing the pancreas now is hard, and taking a biopsy is dangerous, and it's really hopeful now that in Vicky's lab we can engineer things that'll help with diabetes research. But is there something specific that we should be doing? I wonder.
It's a question we can pose, guys, in the chat box. Please let us know. Sarah, Vicky, James, what can we do to get involved in diabetes research? You guys have and projects you've told us tonight. You've shared the fantastic Type 1 Grand Challenge under the Steve Morgan Foundation. Thank you to Liam at the start of the evening for telling us a little bit about the foundation. And I just wanted to close with a few slides from me and Mary in Diabetes Chat today. Here we go. So obviously today we have had our research event. We were looking at the Type 1 Grand Challenge. We've had 3 fantastic researchers and Liam from the Steve Morgan Foundation. If you want to go find out more, please head over to the Grand Challenge website. The link there, you can find out more about the challenge, how it started. You can learn about Steve Morgan and the fantastic team and all the different stories as well, which is fantastic. Like I said, you can learn more about the Grand Challenge senior researchers. So today we've had James, Vicky, and Sarah that have come on. So please go check out their fantastic research projects. I think I've just saw a message. Will this be recorded? Is it recorded? Can I watch it later? Yes, you can, Hayden. So on our website, www.diabetieschat.net/Grand Challenge, you can watch straight away once the stream ends. And please share it out on your social medias, get it out there, and let people know about this fantastic Grand Challenge. And then they're the researchers that we've had tonight that have come on and shared a little bit about the different research projects. So again, go check out more information. And just from our point as well, from Diabetes Chat, please follow us on Twitter. We are also on Instagram and YouTube. And
just to say, Sarah's coming in the chat, and she said that there's a UK initiative called QUOD where pancreases can be donated for research for individuals with diabetes. You just sign up to be an organ donor.
Oh, wow. Wow, that is... Thank you. Please send us a link and we can share it out to the community. Let's have a look. A couple of other final messages coming through from what we can see. Thank you to all the Grand Challenge presenters, amazing insight, and I've gained a lot of knowledge tonight about my T1D and what the future might hold for the younger generation of Type 1 sufferers. Absolutely, I can really relate to that. And thank you, Steve, for your comment. Let's have a look. Mark says, these talks were just like what was said at Diabetes UK in the Grand Challenge event there. Absolutely. And what we wanted to do, our community, when we had the offer to host this, was bring it to the community and to people living with diabetes or have a connection to diabetes. And DUKPC was absolutely fantastic. And is
only a small few of us were there.
Absolutely. And the DDoc Voices were lucky to be there to pay it forward and bring the messages across forward. But tonight we'll be able to show you guys a little bit more of the amazing things the Steve Morgan Foundation is all about and the Grand Challenge. Just having a look, see if there's any other...
There's a link. Sarah has a link for that
thank you, Mary. Donor. There we go. So go check it out. That is the link for... That's the link for the Grand Challenge. Thank you, Mark.
Yeah.
And that is the link that Sarah was talking about. So yeah, go check it out. and I think Josie as well from JDRF. There are also great opportunities to take part on the JDRF UK website. Go
Absolutely.
check that out. From us now, just a final few things is thank you to Mary for hosting this evening. Thank you to Josie and Odette from JDRF for all their support in putting on tonight's event, working with myself and Mary here at Diabetes Chat. Thank you to Liam at the Steve Morgan Foundation, and best wishes to Liam and the team, and Steve and Sally at the Steve Morgan Foundation. And we also want to say thank you to our fantastic researchers tonight for giving their time to talk to you guys, the community, the online community here. So I hope that you find it helpful. The chat will be on for the next couple of minutes, so you can stick around and talk if you want. And then please go share the stream and get it out there, and hopefully more people can watch it and hear about all this fantastic research projects. Mary, anything from you two to finish tonight? What was your take from tonight? What did you really enjoy?
I really enjoyed how it all built together. How like you could see it all like all the little bits of if somebody had a question, it was going to be partially answered later on. I knew it was going to be partially answered later on as well, and it's just you can see how the collaboration is coming together, and it feels so hopeful. I loved the panel discussion at the start, because that was I think that was inspirational, actually, and not just for where it's going and what we will hopefully get out of it, but what new research will come from just even these little starts.
Yes, absolutely. I think it's been really, really great, really insightful. And I wanted to actually share one final thing, if I can share my screen. Let's have a look, see if I can do this. And this was Steve on Twitter, and he said that it's not just him watching along to Diabetes Chat tonight, hashtag Diabetes Chat research event about the Type 1 Grand Challenge, but his partner in Pancreas Crime also watching just before bedtime, and it's a skill night after all. So how amazing that tonight is. The event has helped so many people living with diabetes, parents, young people, and we've reached across the globe tonight, which has been really spectacular. So thank you so much. It has been an absolute pleasure to host this evening, and we hope that you've taken a lot from this evening. Have a great evening, and from myself and Mary and the whole Diabetes Chat team, thank you to the Type 1 Grand Challenge, and definitely go check it out, and we'll see you soon. Bye guys.
Cheerio.